I understand that we're still waiting a couple of minutes for.
Yep. So I'm just getting the live stream to start, which
We
is.
have 3 minutes more.
And then I need to send a tweet about the talk.
What's your Twitter account so I could follow you?
It's African Physics.
I think I'm following you already, but I'll check.
Yep. What's your Twitter handle, real quick?
So it's it's actually on my first slide, so you can see it.
Oh, okay. All right.
Seems we have more participants from the afternoon session than the morning session.
Yeah.
Probably the time differences.
Well, being the first talk of the day is never great because the people just float in.
Well, it's my responsibility to wake up everyone.
Right. I'm really looking forward to this talk.
Mm-hmm.
So this Namib Desert, is it actually in Namibia or kind of?
Yeah, it is in Namibia. It's on the western coast. Well, there's only western coast, but yeah, it's along the coast.
Sorry, Michal, it's your last name Gwizdala. Did I pronounce it very well?
That's good enough, yes.
So it's 9 o'clock. Just give 1 minute, then we start. Okay, so this is day 4 of the Biophysics in Africa conference. The title of this session is Molecular Biophysics. And the first speaker is from the University of Pretoria, Michal Gwizdala. He's going to be talking about photosynthesis underneath stones in the Namib Desert. Michal, the floor is yours.
Thank you for your introduction, and thank you for giving me the chance to actually present my other part of my work, something that we started quite recently with Chart Kruger to understand photosynthesis in nature. So not only in the laboratory, but to go out from our lab and explore it in the nature. I'm going to talk about our very first attempt to do it and share with you some exciting results that were still not published. So I did this work with Charles Kruger, who is the head of the biophysics group at the University of Pretoria. Typically, we stay in this room. Typically, the light is off, and typically there's less people in this room when we do experiments. But as I said, sometimes we try to go out and see photosynthesis taking place in real life. So apart from Chart and myself, also a group of Professor Don Cowan was involved in this project, as well as researchers from the Gobabeb Research Center in Namibia. So in biophysics group, in general, we study photosynthesis, which is an energy converting mechanism that takes place in cells. And it converts one type of energy into another, just like a windmill that converts kinetic energy of wind into energy of potential energy of water. Photosynthesis turns sunlight, so solar energy, into chemical energy that is available for chemical or metabolical processes in the cells. Photosynthesis obviously faces some challenges because the inputs, the conditions change all the time, and also the cells have different needs depending on their state, on what they are doing basically, and when they are. And this is what I'm going to talk about mostly. My personal focus is on photosynthesis in cyanobacteria. Cyanobacteria are unicellular, prokaryotic organisms that are photoautotrophs, so they do photosynthesis similarly to plants. You can see a cell here, a dividing cell, a picture of a dividing cell. They do oxygenic photosynthesis. If you look inside the cell, you can see that there are some structures in the cells. These are thylakoid membranes, just like in chloroplasts. And if you look closer at those thylakoid membranes, you will see that there are large light harvesting complexes sitting on top of the membrane. These are called phycobilisomes. And there are different complexes of the photosynthetic apparatus inside the thylakoid membrane, like reaction centers, cytochromes, and they are involved in triggering electron transfer, as well as then doing charge separation, triggering electron transfer, and then at the end, creating ATP and NADPH that is used for other processes. And as such, this works very, very well under optimal conditions. Energy is absorbed by light harvesting complexes, by phycobilisomes, transferred down to the reaction centers where charge separation takes place. However, under non-optimal conditions, under stress conditions, this photosynthetic apparatus can actually become a death trap for cyanobacteria. This is particularly stressful or difficult for unicellular organisms that can die because of malfunctioning or overexcited photosynthetic apparatus. So what happens is that when there's too much light coming into the system, reaction centers are overexcited, the electron transfer chain is saturated, over-reduced, and they start generating reactive oxygen species that actually can destroy the photosynthetic apparatus and kill the organism. Obviously, as we know, photosynthetic organisms do survive even stress conditions. So we can assume, or we can guess, that they have some mechanisms that allow them to cope with those stress conditions. And yes, they do have them, and we can divide them roughly into two groups. One is photoadaptation. It's a response to stress conditions that involves a generation or synthesis of new proteins, expression of genes. It's a relatively slow response. It allows organisms to adapt to some conditions and live and survive there. Then there's photoacclimation. This is a type of mechanism that is much faster. It's a response at the physiological level, you could say. So cells use what they have at the given time to respond to a stress factor. I'm going to present some of those now very briefly. So the major photoacclimatory mechanism in cyanobacteria involves orange carotenoid protein. I mentioned this protein already on Monday. It's a photoactive protein that, when photoactivated, can bind to a light harvesting complex, to a phycobilisome, and start safe dissipation of excess energy in the form of heat. We can observe it as a quenching of fluorescence. And also there's another protein, fluorescence recovery protein, that can detach OCP from a phycobilisome. And by that, a phycobilisome can then regain its full capacity of sending energy to the reaction centers to run photosynthesis. Then another potentially photoacclimatory mechanism is related to blinking. This is something I discussed in detail on Monday. So apparently phycobilisms have an intrinsic way of dissipating excessive energy when there's too much light entering the system. And also there's a mechanism or mechanisms called state transitions. It's a process by which cells can redistribute excitation energy between two photosystems that are in the photosynthetic apparatus, and by that also photoprotect themselves. The exact mechanisms, molecular mechanisms of state transitions is still not known for cyanobacteria, but we know they can do it. Then when it comes to photoadaptation, there are two very, let's say, interesting mechanisms that cyanobacteria use to adapt to different conditions. So first one is called far-red light photoacclimation, FARLIP, and it's a mechanism, it's a response that allows cyanobacteria to actually grow under far red or even infrared radiation, under far light or infrared radiation. And it involves rearrangement of phycobilisms, so they get simplified. It involves a synthesis of new chlorophylls, chlorophyll D and F. These are chlorophylls that are uniquely present in cyanobacteria only when cyanobacteria grow under far red light. Typically, cyanobacteria would only have chlorophyll A. Then, under these conditions, photosystem 1 is a bit less efficient, but it allows still this adaptation allows cyanobacteria to live or strive and survive under far red light. Then there's another type of adaptation, which is called complementary chromatic adaptations. And some cyanobacteria can do it, so they can exchange pigments in their light harvesting complexes to those red phycoerythrines that can absorb green light and allow cyanobacteria to live under green light. So we study mostly photoacclimatory processes, mostly study them in the lab, but as I said, we are now trying to go out into the field and try to experience photosynthesis and see it with our own eyes in real life. And well, by looking at this picture, you could see, you could say that we didn't choose the best place to see photosynthesis because there's not much happening here. There's basically the sand dunes. Here you can see the Gobab Research Center where we actually stayed during our expeditions. And we do some of our experiments in beautiful places like that. So we are very lucky to see something more than a dark room when doing those experiments. As you can see, this is really located in the desert where we are. This is not far from the coast, so there's some humidity still entering the desert. This is around 2 hours drive from the coast. So if we look closer, you could really ask the question whether there's any life there. It is a desert. There's basically sand and stones. And it's not surprising that we don't see too much life because it's extremely arid. There's not much water coming into this part of the desert. The temperatures are very high. This is something that we experienced while doing experiments in the field. And there's a lot of light. So there are no trees, no objects that could give us a bit of shadow shade, typically. And so, yeah, we could ask whether there's any life. And life is there, but life is a bit hidden. So if you pick up any of those rocks that you saw on the picture, you have a high chance of seeing quite rich green biofilm growing underneath those stones. So I'm talking about translucent stones. These are quartz stones typically. And you could see that there is a grow of quite complicated biofilm there, composed of many different types of organisms. But what we are obviously interested in are photoautotrophs. So we know that there are cyanobacteria there, and altogether stone plus those cyanobacteria form something that we call hypolis, or is called hypolis. So our goal now was to understand how cyanobacteria adapt and how they photosynthesize, how they manage to actually survive those extreme conditions while living underneath the rock when they have really much less light than maybe needed for photosynthesis. So first thing to understand was to understand photosynthesis in hypolis, we wanted to understand the conditions that they experience. So first thing obviously was to look at the stones. That's the easiest thing you can do. You leave the stone, and what you can do, you can measure how deeply into the soil you find green biomass. So we would find that we can get biomass up to 8 centimeters below the soil level, and that corresponds to 0.005% of photosynthetic active radiation. So that's a very, very low, small amount of light. However, we also found cyanobacteria growing underneath the stones to up to 17%. So you can see this is more than 4 orders of magnitude span of light intensities that cyanobacteria experience underneath the stones. And this is quite spectacular as a result. So this is something obviously to look into more into details to see whether there are different adaptations at different depths at different light intensities. On average, light transmission is around at 3% of photosynthetic active radiation. So that's quite comfortable. These are quite comfortable conditions when it comes to light. So here you can see a spectrum of solar radiation measured in GOBABEP. And here you can see transmission through the stones. If we normalize those peaks, you can see in blue and red some transmission spectra of some stones that we actually collected. What you can see as a common feature of those two spectra that I'm showing here is that blue light and UV is completely attenuated. So that's actually very good news for any organism that lives underneath the stone. They don't get this most stressful part of radiation. You can see that in both cases, the radiation is also relatively redshifted. So I'm showing here only 2 spectra, but actually we would observe a bunch of different spectra, all of them, however, falling between the blue and red. So the blue and red spectra here are somehow the limit, the most blue one and the most redshifted spectrum that we managed to find, typical spectrum that we managed to find. So one other conclusion that we also have from those measurements is that you cannot just judge or guess the spectrum by looking at the stone. So sometimes the color can be misleading and you cannot really tell what would be the spectrum exactly. One other interesting feature that also would probably promote some additional experiments and measurements is that if you look at the difference between those 2 extreme spectra, you can see it here in gray. This difference overlaps with absorption spectrum of photosynthetic pigments, so chlorophyll A, carotenoids, and phycobility proteins. And that would tell us that the color of the stone could actually be also a driving force for some specific adaptations to different conditions. That's something, again, that we'll have to look into in the future. So now, knowing a lot about the stones, the next step was to try to understand a bit the composition of the photosynthetic apparatus. And we decided to approach it first by doing methanol extractions. And when we took absorption spectra of those extracted pigments, we could see that very often it was enough for us to fit chlorophyll A spectrum and measured as well carotenoid spectrum to get a perfect fit of measured total pigment extraction spectrum. That tells us quite a lot. That tells us already that there are no chlorophyll B and C in the extracts. And that means that there are no eukaryotic algae present. So we are talking only about cyanobacteria here. Interestingly, given the fact that the transmission spectra of the stones are relatively redshifted, we also didn't see any chlorophyll D or F, which would be involved in far leap if there was any far leap. So that was very interesting that cyanobacteria do not really use this strategy to underneath the stones. However, that was not the end of the story with the spectra. We also observed that often we needed to add another component to our feed to be able to reproduce the spectra. And this component was cytonemine. Cytonemine is an extracellular pigment that helps cyanobacteria to cope with UV. And that's very intriguing. Why cyanobacteria would produce and accumulate cytonemine if we know that there is basically no UV in hypolis. So that's a mystery. That's again something that it's an open question it's a question that we now ask, and we'll look for the answer in the future. So now I would like to move to real measurements of fluorescence activity or potential for fluorescence activity. Here we use photosynthetic activity, sorry. Here we use fluorescence parameters, so we could measure directly in the desert some fluorescence emission parameters to be able to tell something about the photosynthetic activity of the hypolis. So first, if you look here, we went out into the desert and we were lifting random stones in the desert, and we would measure FV over FM. And high values of FV over FM in general mean that those hypolis are photosynthetically active. and low values mean that hypolysts are photosynthetically inactive somehow. They are quenched or photosynthesis is just switched off. So you can see we did see a whole range of different stones with different values of FV over Fm. But then when we left those stones for a bit more than half an hour, still in the dark, we observed that FV over Fm dropped for most, actually all of the stones that we looked at. So since this was not related to any light stress or quenching induced by high light, the only factor that actually changed is the amount of water. So here we could see that water most likely induced this quenching of fluorescence, so, and by that the inactivation or quenching of photosynthesis. We are kind of intrigued by that, so we decided to look more into detail. We established a very simple humidity chamber in which we took and we put inside those stones that have very low FV over Fm values. And after a certain amount of time, we observed that some stones would actually increase their FV over Fm values, meaning that actually water availability is a stress factor for photosynthesis and it regulates photosynthesis. So when there is water, there is photosynthesis. When there is no water, there is no photosynthesis. Interestingly, when we then took those photosynthetically active hypolysts outside and expose them to full sunlight. By full sunlight, I mean sunlight that is filtered through the stone. We observe absolutely no change in FV over Fm. And what tells us is that even at the maximum light that hypolysts can get, they do not experience any light stress, or at least we don't see it in our measurement. So we could ask the question, why we don't see any light stress? And one reason would be, or why we don't see any difference in fluorescence. One reason could be that there is no orange carotenoid protein, but the other reason could be that there is no photoinhibition, so there's no light-induced damage to the system. And to answer this question, we actually used a metagenomic tool provided by our collaborators from Professor Don Cohen lab. And we could look for OCP genes in the metagenome of hypolysts from the Namib Desert. And interestingly, there is no OCP1. So there's no type of OCP that is responsible for photoprotection. So indeed, that confirms that light is not a stress factor for hypolysts in the desert. However, we found genes that are evolutionarily correlated with OCP. These are genes called HCPs and OCPX. And while their function is still not very clear, it was suggested in a couple of articles that they are involved in desiccation tolerance. So that's another point supporting our idea that water is a major stress factor and regulates photosynthetic activity. Having this metagenomic tool in hand, we also ask another question. So I told you that we didn't see any chlorophyll DNF. So we really wanted to check whether it's because we didn't see it because our experiment in the desert was not accurate enough or not sensitive enough. So we decided to look for markers of Farley. And one of the markers is APCE2 gene. And this gene was, again, no present in hypolysts. So that confirms our biochemical characterization that Farley is not a major strategy that cyanobacteria would use to adapt to hypolysts. We interestingly found 20 genes that are similar to some linker proteins of phycobilisomes. That tells us that there's still a lot of diversity to explore, especially in those extreme environments. And finally, we also didn't see phycoerythrin. Phycoerythrin is this red pigment that is used in complementary chromatic adaptation. So it means that if there's no phycoerythrin, cyanobacteria occupying hypolysts also don't use complementary chromatic adaptation. So they don't use Farley and they don't use complementary chromatic adaptations to respond or to adapt to the conditions. Overall, to conclude, the general conclusion is that cyanobacteria in extreme environments occupy less extreme niches. So by less extreme, I mean those niches like underneath the stones when they do not experience excessive irradiation, they do not experience UV, and possibly actually those life conditions also can reflect some adaptatory mechanisms that we still have to explore. One other conclusion is that water is critical, and here actually the stones also provide benefits because stones as such can help accumulate water. Also, the extracellular polysaccharides that form the biofilm, they are known to accumulate water, so they allow for photosynthetic activity for longer then. Interestingly, there are no other photoautotrophs in hypolites other than cyanobacteria in the Namib Desert. And cyanobacteria don't use photoadaptory mechanisms like FARLIP or CCA. At the end, I would like to thank the biophysics group, in particular Chart, then our collaborators from the UP, Pedro and Don, collaborators from Gobabep, then Min Chen and Peter Richter, who provided us with reference spectra that we use for fitting, as well as Diana and Aaron for helpful discussions. Finally, I would like to thank University of Pretoria, NRF, Cloud Leon Foundation for funding, and thank you for your attention.
Thank you very much, Michael, for this very, very interesting talk and very nice presentation. The floor is open for a couple of questions. So if you have a question, just unmute yourself and ask, please.
I have a question. I still cannot find a raised hand thing, but are there in the photosynthetic bacteria that you found, are there metals that are part of the process? And are those metals provided by, are they leached into the soil somehow and get?
I'm sorry, I didn't hear the word that you, are they what? Are there what?
Are there any metals that are involved? Metal compounds?
Oh yeah, obviously, like, so yeah, in photosynthesis. So photosynthesis is driven by pigment protein complexes, and many of those pigments use metal atoms as a part of the pigment. So there's iron, there is magnesium, manganese, calcium. All of this you find in the photosynthetic apparatus. There's also copper, sometimes in cyanobacteria. So there's quite a lot of metal, but these are not large amounts. I don't know if it answers your question exactly, but.
Well, I was wondering, well, like, what's the relationship between the geology of the stones and the metals that are used in the photosynthesis?
The way we approach it now is that actually the geology of the stone is reflected in its optical properties, and that has the biggest impact. I doubt that actually the availability of metals would be somehow an important factor for limiting photosynthesis in those conditions. So all of the metals that are needed for photosynthesis are there anyway in the soil available. So I'm guessing that actually composition of the stone then is reflected in those transmission spectra that I showed, and that's what's important. But none of us in the team is a geologist right now, so this is something exciting, however, beyond our expertise right now.
Thank you.
So are there any other questions? Please ask. Since there is none, thank you very much, Michael, for this great talk.
Thank you.
We now move to the next speaker, who is from the Kwame Nkrumah University of Science and Technology. She's also a member of BioStrong Africa, and she's the one representing BioStrong Africa in Ghana. She's the coordinator in Ghana. So she's going to be talking about triggering receptor expressed on myeloid cells and cerebral malaria pathogenesis. So Amma, if you're ready, please proceed.
Thank you so much. I know my talk is not going to be strictly biophysics, but I hope you will enjoy it. As Emmanuel has already introduced me, I'm talking about triggering receptor expressed on myeloid cells, or TREM1, and cerebral malaria pathogenesis. Sorry, my screen is stuck. So, okay. Malaria continues to be a major health problem. A lot of research is in this area, but currently we still have 229 million malaria cases with 409,000 deaths globally annually. So cerebral malaria is actually a severe form of malaria. It is caused by Plasmodium falciparum, and most of the malaria-related death is caused by this type of malaria. It's malaria of the brain, and it is usually found in children under 5 years in sub-Saharan Africa. Cerebral malaria is multifactorial. A lot of processes come together for the disease to be established. We have endothelial dysfunction. We have dysfunction in the pro-inflammatory cytokine, both in the production as well as regulation. Then parasites attack red blood cells, and these parasitized red blood cells are sequestered in the endothelium, leading to other complications implicated in cerebral malaria pathogenesis. So when the parasite enters the body, it leads to what we call alterations in coagulation, and it releases certain proteins such as histidine-rich proteins, and these proteins lead to a loss in the barrier, the blood-brain barrier, and it leads to impaired vasodilation. Host factors are activated, pro-inflammatory cytokines are made, and there's expressions of other proteins and cells. The parasitized red blood cells are sequestered in the endothelium, as I've already said, and that leads to what we call microvascular obstruction. When this happens during the infection, cells that express TREM-like 1 or TREM1 are stimulated. These cells then, such as monocytes, such as neutrophils, all these express TREM. So these TREM cells stimulate the release of chemokines, and they also trigger the expression of pro-inflammatory cytokines such as TNF1. When these cytokines are activated, it is believed that it contributes to the damage of the endothelium instead of the repair, because such individuals do not make enough TREM-like transcript 1 protein, which is a TLT1 protein. And this is supposed to give inhibitory signals. And it is believed that this inhibitory signals does not happen, and hence anti-inflammatory cytokines are not produced. And so TREM is potentiating inflammation, but the anti-inflammatory signals are not produced. We have what we call EPC, or endothelial progenitor cells. The function of these cells is to repair the damage caused by the sequestration of the parasitized red blood cells on the endothelium. And the function of damage also releases what we call CEC, or circulating endothelial cells. These are markers of damage. So the aim of the study was actually to determine if there's any interaction between TREM1 and microvascular damage or repair as seen in CNS physiology. So our objective was actually, the first one was to compare TREM1 and TREM-like 1 biomarkers in pediatric cerebral malaria cohort patients. And we also had those who we call uncomplicated malaria. That is the non-normal malaria, as we would say, that does not lead to severe disease. And also to determine if there's association between the microvascular damage experienced in cerebral malaria patients and the results, and if this is based on endothelial integrity. The study was a longitudinal study, and we recruited patients from the age of 2 to 12 years. And we had 3 main categories. We had cerebral malaria cohort, we had what we call the uncomplicated malaria cohort, and we had the healthy controls who did not have malaria and who did not have any diseases going on at the time of recruitment. We collected the samples at 4 different time points for cerebral malaria, because at the baseline, or day 0, as we call it, the cerebral malaria patients are in coma. So we take that sample when they're in coma, we take their blood as well as we take their cerebrospinal fluid. And then when they wake up from the coma, we call it recovery. We take samples at that time, just their blood. And then 7 days post-recovery, 7 days after they recover from the coma, we take their blood, and 14 days. But for the uncomplicated malaria or normal malaria, we just come at recruitment. We take their blood, which is day 0, when they come with the malaria parasite. And then 7 days after we take the first sample, we take day 7 and then day 4. And we do same for the healthy control, those who do not have malaria or any known diseases. We did this study at the capital of Ghana, which is Accra, and we collected the samples from 5 different hospitals, which are children's hospitals in Accra. For our blood samples, at any of these time points, day 0, recovery, we took 5 ml of the blood sample, under which we use some for microscopy, we use some for full blood count, we use some for flow cytometric analysis, where we looked at the monocyte, neutrophils, among other cells. We also did bacteremia to rule out contamination with bacteria. We then did ELISAs to look at our levels of TREM and other cells. So let me share a few results that we had so far. For the soluble TREM1, what we noticed was that individuals who have cerebral malaria had higher levels of TREM1 compared to the anti-inflammatory TREM-like transcript 1 protein. They had lower levels. And uncomplicated malaria individuals had higher anti-inflammatory TREM-like transcript 1 protein compared to TREM1, which is inflammatory. So what this shows us is that from cerebral malaria cohort individuals, TREM1 was actually important in the driving of the pathogenesis of the disease. Because of this, we wanted to find out, can we categorize our patients into microvascular damage and repair? Let me take you back a bit to tell you that when there's sequestration of the prioritized red blood cells, it leads to damage of the microvasculature or the microvascular. So when this happens, the body makes endothelial progenitor cells, and the function of these cells is to come and repair that damage. And the size of the damage also leads to production of circulating endothelial cells, and these are biomarkers of damage. So we wanted to come up with a cut-off point value by which we can say that yes, an individual has damage or has this amount of repair. So we use this procedure and then use the Gaussian mixture model to come up with a cut-off value. And the cut-off value was 0.1912. And this value, all values that were below the cutoff point were categorized as repair, and those that were above the cutoff value were categorized as damaged. Based on this information, we used it to stratify our cerebral malaria cohort group and our uncomplicated malaria group. What we noticed was that the cerebral malaria patients had a higher damage, about 74% damage, with only 25% to repair. And the uncomplicated malaria group were able to repair most of their damage, had about 70% to repair, as compared to only 29.1% damage. What this shows is that damage actually plays a role, or inability to repair the damage of the endothelium makes an individual prone to develop severe disease as compared to uncomplicated disease. So what we can say here is that soluble TREM1 is important in disease severity of cerebral malaria, and endothelial progenitor cells are crucial in recovery from cerebral malaria. So individuals who, I didn't show that data, but that we have data that shows that individuals that have cerebral malaria have very low levels of endothelial progenitor cells, and hence are not able to repair their damage. And this is what causes them to have severe disease. And those of our patients who died had even lower endothelial progenitor cells compared to those that recovered. This supports the theory that endothelial progenitor cells are crucial in recovery of cerebral malaria. Again, we can say that endothelial damage and repair are key in cerebral malaria. Damage occurs, but the ability of the individual to repair that damage will be good for survival of that cerebral malaria. So biomarkers whose activation or secretion move more towards repair than damage enhances endothelial function. And those that move more towards damage than repair enhances endothelial dysfunction. I'd like to thank the funding agents. We got the grants from NIH and the collaborating hospitals, as well as the team and Emmanuel and BioStract Africa. Thank you very much for your attention.
Yeah, thank you very much, Amma, for this great presentation. We know malaria is still a serious problem in Africa. The death rate is still very high, and this has a major impact on families, communities, and the nation's daily livelihood. So thank you very much for this presentation. Is there any question from the audience?
Well, I was wondering, it seems like, so the phenomena that you elucidated might be mediated by mechanical properties of the cell. Do you think, does that sound reasonable? And then what would that mechanism be?
So when the sequestered red blood cell is trapped at the endothelium, what happens is that the first cells that are recruited to that site are neutrophils, and it also brings what we call stromal SDF1 is also recruited. And this SDF1 then signals to the endothelial progenitor cells, which are produced in the bone marrows, that there is a damage at that site of injury. And so the EPC or endothelial progenitor cells should be recruited to repair the damage at the local site. So yes, the cells at the site have a big role to play in the disease.
So Amma, just as an aside, there's been little success in terms of vaccine development for malaria. What could be, do you have any idea what could be the reason for this? Most of the vaccines have not been able to offer full protection for the disease. Can you shed light on this?
Yes, so basically malaria has a lot of antigens at play in the disease. That is why immunity has been, protective immunity has been a challenge because several antigens have a role to play. So today, and in this antigen will be presented to somebody, the person will build. Another time, the person gets malaria, another antigen is playing a role. So I think that that has been one of the main reasons why vaccine development has been a challenge. But beside that, for us, those of us in Africa here, I have had bouts of malaria, but what you've noticed that as an individual grows, the episodes reduces. Even though you are not protected permanently, because I still occasionally get malaria, the episodes compared to a child is actually much reduced. So it means that we have to find out how do we make protective immunity last longer. That will be key to getting a good vaccine for malaria.
Yeah, some researchers also suggest that it might have to be a successful vaccine must be something that could target multiple antigens together. Yeah. Yeah.
That's true.
Yeah. So thank you very much.
Thank you.
Is there any more questions? So if not, that brings us to the end of this cellular biophysics session. I don't know if Simon, do you have anything to comment? Or Lawrence?
Nope, just the afternoon session starts at 15.
15:00 GMT +2?
Correct.
All right. So thank you everybody for listening and for the two speakers. Let's give them a round of applause. All right, see you everybody. Bye.
Okay, see you this afternoon.